I'm the founder and CEO of ADVA Biotechnology. Been in this field for about 20 years now, since my PhD, through different positions in the industry, both in medical device industries, but mostly cell and gene therapy. Been active in a lot of societies like ICT, ARM, and then a few others. So I think my first entrepreneurship project in this field was basically my PhD. 20 years ago, we're trying to differentiate MSCs, mesenchymal stem cells, into insulin-producing cells using genetic modifications. So it wasn't immunotherapy, but it was the same technologies, same techniques, the anti-vectors, gene therapy into cells. So that was the first touch in that industry, and since then, been doing it for 20 years. Yeah, so in ADVA Bio, basically what we did is we identified, like many others, that there's a very critical bottleneck. That's the manufacturing of autologous cell therapies. It's a very complicated manufacturing process, very labor and very highly qualified labor that is needed. In order to scale this and really get this to patient, you need to have an automated, scalable manufacturing device. And that's what our aim was. Try to develop, or we've developed, a technology that basically automates and puts the artificial intelligence behind that system in order to enable highly skilled manufacturing in a very simple and automated manner. That's a very good question, because that was part of the idea behind the device. Developing a device for something that exists is usually what you do. So there's a process, there's a protocol, and then you automate that. But that will never work. We have hundreds of different immune cell therapies that are developed, and each one is a little different than the other. It could be different in the media, could be different in a few steps in the process, the vectors they use, the supplements. So you cannot really develop post-mortem, because it will be post-mortem. They will die before you get an automation. So we needed to develop something that is very flexible and will enable all that flexibility in order to allow the scientists to develop their therapies. So the simple question to your answer is yes, it's very flexible and it's an open platform. So you can use whatever media you want, whatever type of cells, whatever process, and program that into the system. The media is much more than just a mixture of juices that allows expansion. It's not only how fast the cells grow, but it's their quality. The media has in it a lot of different supplements, a lot of different growth factors, and we know that cells react to them. So when you choose your media, you have to look not only on what is the number that comes out, but what is the effect on your phenotype. So how does it affect the different types of cells in it? How does it affect the important or your critical quality attributes and potencies? So if you're looking at earlier stem cells that you want to maintain that phenotype, or more towards the killer phenotype, for example, in T cells. So it's a very complicated matrix, and people have to understand that there's a lot of variations there. And you would like to take to try to find the media which is the most reproducible for different donors. And that's quite a difficult challenge. Definitely. I would point out two major differences or important issues. One, serum, doesn't matter if it comes from animal or from human, is a risk. It's a risk factor and has to be mitigated. So that's one issue. If you can find something to reduce that risk, meaning chemically defined, it's always better. Second thing is, what I mentioned earlier, is that variation. When you do media, again, doesn't matter, serum, doesn't matter if it's bovine or human, it's a pool of different serums. Once it's a pool, every pool is different. Trying to prepare big batches, to try to eliminate that, but there's a difference. So the amount of growth factors, the amount of cytokines, the level of cytokines will be different. And this is something that you cannot measure quantitatively as part of the manufacturing, can't really control it. So you need to try, that the best opportunity, the best solution would be trying to find a chemically defined, so minimal serum, trying to have whatever you have in that soup to be as defined as possible. So I think the major challenge was exactly your first question, was how do you develop a device for a developing industry that doesn't even know what's their next step. So you need to develop a device that will fit as much as possible, wide as much as possible on the one hand, and on the other hand would be specific as much as possible. So we needed to think differently. Again, not going from the end to the start, but trying to think where will this head and what will be the critical parameters that we want to have in these in this device, meaning what do we want to control, what would be the range of cells that you want to start with, what would be the range of cells that you want to end with, what are these unit operations that you need to do, and how could you build a device that has all these parts, and practically you build your own puzzle out of that. I think that was the biggest challenge, which took us to a point where we had to reinvent the way we grow cells. So we're not growing them on culture, we're not growing them in a stir tank or a dish, a culture dish, not a stir tank, not any variation of that. It's a continuous flow culture, a uniform, unidirectional flow. Totally different than the way that we're used to, but that was the only way to enable exactly these parameters and exactly these challenges, to come out with a system that is flexible and very, very open to changes. Okay, so I think people theoretically look at allogeneic and say that's the solution. But there's two issues here, or three. One of which is nobody really could expand the cells and maintain their phenotype and activity to the numbers that we're dreaming. So taking from a single donor a few T cells, engineering them, and getting them to the numbers that we're talking is still a challenge. Once you pass that challenge, there's the safety regulatory challenge. So we are taking these T cells and we're eliminating their immune reaction, for example, or their immune identity. And theoretically it's safe. But why am I saying theoretically? Because we're starting with a few million and we can maybe eliminate and test them, and getting to tens of billions or hundreds of billions. We need just one cell to get back its immunity in order to be a danger to the patients. So the regulatory burden and the safety burden to approve this would be much higher than people expect. Because we looked at T cells, at CAR Ts, and said, oh, this is very simple. Phase 2 and it's approved. Because we're risking one very sick patient with his own cells and not risking now a few hundred or a few thousand patients with that same batch. So the regulatory burden is going to be very challenging. The second challenge would be the amount of cells that you need to get to, which is still not really doable. And the third challenge would be then a technical challenge of building a tool to manufacture these cells. So only once we pass these three barriers, autologous allogeneic could really reach the market. And then there's the question on things like cancer. It is a very, very diverse disease. So there's no one drug that can work on it. So it's much more personal in characteristics than it is allogeneic. So you could look at leukemias, CD19, yes, that might be allogeneic. If we're looking at very specific markers and TCRs, that is not going to give a full solution. It might give a partial solution, but not a full. So I don't see allogeneic becoming a true and viable therapy, cost-effective and large-scale in the upcoming near future. It will take a lot of years to get to that point. We will get to that. But to get to that point, so I do believe autologous has a very big market in the next 15 years easily. So I think if you look at cell therapy in general, even at CAR Ts, etc., we're going back to the autologous side of it. So the allogeneic, and that is very high level, haven't shown enough efficacy. There is transfer efficacy, there are some efficacies, but it's not dramatic as we saw in autologous. Autologous works in general means, especially around immune cell therapies. So if you're looking at that arena, there's a huge market there, there's a huge potential. Not only cancer, but viruses, HIV, hepatitis, things like that is a big challenge for humanity today. CAR Ts could work. If you're looking at autoimmune diseases, we can basically educate the immune system. The only issue here is cost and availability. So once we pass that point of making it simple to manufacture and cost effective, I think cell therapy would become a huge market and a really significant portion of medicine as we know it. Okay, so as we see the growth or expansion of this technology, it's a stepwise. It will start probably going to companies just to make their manufacturing efficient. Because today, if they're manufacturing at a small scale or very cost effective, very uncost effective, if you put a few devices in the same room, you can upscale your manufacturing and make it more cost effective. On the other hand, there's the hospitals that are, again, really at this point are limited. Hospitals, academia, that are starting to develop these tools, and they have a limit of how many they can do and the quality of GMP that they can come out with. So I see it expanding in two paths. One, going to these companies and helping them become more efficient and more cost effective and grow into market, and the other is helping these hospitals. Maybe one day these two will merge and we will have a manufacturing of companies at point of care. But this is an evaluation, the market and the regulatory has to evolve into that point. But in both sides of the industry, we do see value to such a technology that will enable these therapies to get to patients. So I think exactly what I said. So if we have this technology, then now I can put on a table in a pharmacy in a hospital and manufacture a personalized cell therapy that is exactly personal and adapted to the patient's needs at a cost-effective and simple manner. That will be a game changer. That will be an enabling technology for the next generation of personalized medicine. And what we're seeing ourselves as a company is not a tool developer. It's a platform, an enabling platform to really change the way the industry and change medicine as we know it today.